So this is Jenny LeRae with Research America, and welcome to all of you who have joined us from literally around the country. I want to thank both pharma and the American Society for Microbiology for their support of this webinar. Research America's goal for this session is to better understand the key approaches underway for the development of COVID-19 vaccines and treatments. We'll also learn more about some of the key partnerships between government, academia, and industry that are intended to improve and speed up the R&D process. Serving as our moderator and hopefully jumping in as well as a panelist, and don't worry, he's adept at doing both, is Dr. Mark McClellan, former FDA commissioner and currently the director of the Duke-Margolis Center for Health Policy. And we're also pleased to have him on the Research America board. We're also joined by two other incredibly knowledgeable guests, Dr. Stefano Bertuzzi, CEO of the American Society for Microbiology, and Dr. Julie Gerberding, former director of the CDC and now Executive Vice President and Chief Patient Officer at Merck. Before I hand the reins over to Mark, two housekeeping items. By now, I think we're all used to online sessions, but as a reminder, please submit your questions via the question box. If you've connected to us by phone, you can unmute yourself by pressing *6. The session will be recorded, and we will be sharing that with all of you. But if you have colleagues who weren't able to join us or who are coming in late, please let them know that they'll be able to access the recording. Okay, Mark, well, I think we're ready now for you to take it away.
Great, Jenny, thanks for that introduction, and all of you who are joining us this afternoon, thanks for being with us today. I'm very pleased to have this opportunity to discuss with Julie Gerberding and Stefano Bertuzzi some of the important current issues in the development of better treatments, better response for the COVID-19 pandemic. As you all know, there is an unprecedented amount of activity across the research ecosystem focused on developing a vaccine, and in the interim, developing more effective therapies for COVID-19. Would also add that it's not just about the therapeutics, it's about innovation on the diagnostic side as well, especially for a virus that can often pass without symptoms to many other individuals. Effective diagnostic testing is understandably front and center in the efforts to combat the pandemic, both here in the United States and around the globe. And many of the efforts that are underway in these areas of biomedical innovation are building upon our experiences and research developed in past epidemics, including MERS and SARS, the previous coronavirus epidemics, and others as well. And we're also seeing some new opportunities for innovation that have come along in more recent years, such as building on developments in areas like monoclonal antibodies. So lots of innovation activity underway. And Julie, I'd like to start off with you. You've been around the block a bit on this. Some of it we've done together. I remember back in the SARS outbreak in 2003 when I was at FDA and you were at CDC. Could you talk a little bit about what's different now versus then in terms of how the research community is responding? Obviously, this is a even more impactful pandemic, but other things have changed as well.
Well, obviously, the first SARS outbreak really was almost the pilot study for what we're experiencing today. That outbreak was characterized by late recognition, or at least relatively late recognition. But once it was clear that it was a very serious pathogen with what proved to be ultimately a 10% mortality rate, I think the science did kick into high gear fairly quickly. We had diagnostic tests available fairly soon, but it, not surprisingly, is basically the case in every emerging outbreak. People try the things they have at hand first. And so, of course, antiviral compounds that were available were rapidly screened. Things that we used for immunotherapies or passive immunization were tried fairly early in the course of the outbreak in Asia. Unfortunately, none of them were studied in any kind of really good controlled way. So at the end of the day, we really didn't have much information about what was effective or might have been effective, et cetera. And of course, vaccine work got started, But I think one of the really, in retrospect, sad things about SARS in 2003 was that none of those things really made it across the finish line. We didn't complete the job, find out what was working or what was most likely candidate to be working for future situations, in part because the disease went away and they became harder to test. But nevertheless, I think there was a loss of continuity there. Fast forward to where we are today, and we're experiencing basically an outbreak of clinical studies looking at all three of those categories again, looking at the existing compounds and screening them very quickly, looking at immune therapies that we have in terms of attacking the cytokine storm and the other immunologic manifestations of the disease. And then, of course, working fast and furious. I think there are more than 70 vaccines in progress. At least 5 of them have already been put into clinical trials of one sort or another. So out of the starting gate, we're exploding with science and scientific opportunities. And in a sense, that's created the paradox of almost starving in the midst of plenty, because there's so much going on, it's going to be very difficult to tease it all out. Or if you're a clinician trying to make a good decision for a patient, where do you turn to know what's the best opportunity and option for the patient that you might be trying to study? So it's good news and bad news, but I think our science has never been more exciting, and I'm sure we're going to find solutions. It's just a question of how fast can we get there.
Yeah, and in terms of how fast, one of the things about vaccines is that they're not that fast. And maybe you could talk a little bit for this audience about why that has been the case. In previous outbreaks, it can take years to develop for new for novel viruses. You mentioned with SARS, the epidemic had kind of died down before the vaccine efforts really got going. But this time around, there are a lot of efforts underway to try to do it faster. Can you maybe talk a little bit about that as well?
It's helpful, I think, to compare what needs to happen in terms of an antiviral treatment versus a vaccine. First of all, with antivirals, we have tons of them on our shelves, and some of them are in clinical practice. And of course, those we're already using and we understand the safety are the first place to look for activity against a new virus. And that's obviously what's being done now. The rapid screening of everything we have in the background on the shelf that looks like it might be a useful compound is certainly well underway. And with antiviral therapy, the endpoints happen very soon. They're clean. The patient either gets better or he or she doesn't. The patient lives or the patient doesn't live. The patient ends up in the unit or doesn't end up in the ICU. So it's faster to do the antiviral work just as a general principle. They're also usually easier to make. Not always. We're seeing some challenges with the synthesis of some of the promising antivirals right now, but generally they're easy to make and relatively inexpensive to produce, and the regulatory pathways are fairly clear. With a vaccine, it's a very different story. Generally, we don't have vaccines sitting in the freezer, although in the future with CEPI and other efforts we might. We have to really do an extensive amount of preclinical and safety work because these are generally new, unproven compounds, and safety is everything. It takes a long time to demonstrate that they protect against infection, and it takes a long time to prove that they're safe. And when you're delivering the vaccines to otherwise completely healthy people, that safety is just absolutely the most important thing. So no shortcuts there. You really have to be sure that we're dealing with a safe product. And then finally, the manufacturing is a very challenging situation, both in terms of the capital investment for producing sterile products, but also the timeline and the regulatory process to get them approved from a compliance and safety perspective. So there are no shortcuts to vaccine development. And while we might be able to get some investigational products into the arms of people sooner than we expect to have them available for the general public, this is not going to be a fast race. It's something that we're going to have to expect and not overpromise on the timeline for, because if we overpromise and underdeliver, that will just be one more hit to our credibility in the process.
Yeah, so longer development process in part because you're often giving vaccines to healthy people or people who are at risk but not infected with the disease. Also because you're waiting for immunity, demonstrating that immunity does develop in people who get the vaccines, and it's enough and lasting enough to make the vaccine effective in practice. So, and you mentioned all the manufacturing issues too. That said, there have been some steps to try to reduce the timeline, and we have Ebola vaccines now that are making a difference. Can you talk a bit, maybe a little bit about that experience and what's different, and maybe how we can build on it today to make more rapid progress while recognizing all of these steps in vaccine development and large-scale production?
Yeah, I think the path to the approved Ebola vaccine contains a lot of lessons. Probably the overarching lesson was the amount of incredible collaboration that it took every step of the way, from the very inception of the vaccine by Public Health Canada with the support of the Canadian Defense Department, how that got brought to the US Defense Department as part of our threat agent activity. And then the role of the small biotech company, New Links in Iowa, and how they did the original preclinical work and developed enough supply to be ready for clinical trials. And then kind of the coincidental recognition on a timeline that there was this dreadful West African outbreak, and the role that Merck took in terms of being able to bring that very promising vaccine forward to scale and with the clinical development speed that really is unheard of in the world of vaccine development. Having said all that, and with the support of many collaborators on the front line, not to mention the governments of Western Africa and all the health workers and the people who volunteered to participate in these clinical studies, the first dose went into a human being in Africa in, I think, 2014, and the final FDA approval did not happen until December of 2019 for a vaccine that was in high need given the new outbreak in the DRC. Everybody was bending over backwards to try and move this vaccine through the regulatory and manufacturing process, and yet it took time. And I think that's really one of the important lessons. How can we shorten that time, especially when the clinical studies need to be done in areas of the world that aren't familiar with clinical trials or informed consent, et cetera, et cetera. We need to have a global system to be able to move these things through that process much more quickly than we really have been able to demonstrate so far.
And what's your sense of how we're doing in getting that better, more collaborative global process in place? It does seem like there have been a lot of efforts underway to align regulatory requirements across agencies all over the globe, and ways to at least think about getting that manufacturing capacity in place, even when products are still at risk, even while the clinical trials are still going on, so there's not a long delay there. How do you say all these activities are going right now?
Well, in the case of this outbreak, this pandemic, I don't think the world has ever been more aligned on the importance of moving these things forward. What the FDA is doing is absolutely fantastic. I think some of the pieces that got put into place before this happened, for example, the liability protections in the United States for manufacturers and people who are stepping forward with antivirals and vaccines, has such a barrier to the ability to confidently invest and study and then deploy innovative products. And that box has been checked for the US. It's still an issue in other parts of the world. So many things have changed and improved. And I think we just need to keep going, because there's still more that we can do. But we can't sacrifice scientific integrity in the process. And I'm worried about that with some of the testing issues. Let Stefano talk about that. But I think also safety, even for the antivirals or the immune therapies, we need controlled clinical studies. And I know that's frustrating to people, but we have to understand the benefits and the risks of what we're doing. Even when it seems like we should just be able to blast out because the disease is so serious, we need to understand and know if we're adding benefit or harming patients in the long run.
Yeah, so more efficiency and speed, but not cutting these important corners in development and production. May come back to some of these issues, but I'd like to bring in Stefano around some of the issues that ASM has been involved with, which I know touches on vaccines and getting vaccines used effectively when they're available, but also a range of other topics. Could you talk a bit about what your members are doing in the COVID-19 response?
Sure. Do you have like a week time? I can I can get through all of that. No, first of all, I was considering while Julie was speaking how good it feels to be here with you today and talk about science and where we came from the first SARS outbreak and where we are today. You know, we owe this to all those people who have suffered, who have died, and who are suffering now. And I think that what we're doing here, this conversation, is really what, to quote Francis Collins, and it's like, you know, the only thing that's more infectious than the virus is the hope that comes from research. And so this is really, I think, a beacon for where our members as ASM really contribute. And first of all, you know, ASM encompasses from the very basic science, which is so important in all this, the basic virology in this. And as Julie was referring to, the fact that we were able to develop so fast therapeutics, repurpose, develop vaccines and all that is because we're building on all that body of research that has happened over time. There are, until December 2019, there were 6 coronaviruses known, right? And of these 6 coronaviruses, 4 are nothing more than a cold that will, at worst, keep you at home 1 day or 2 from work. And 2 were SARS and MERS that had a very different scenario. And they were different in and of itself. If you think about it, one of the reasons why Julie was saying, you know, we didn't take it to the very end, it's also because we were able to contain SARS, right? It was essentially a peak and then it stopped. So it behaved in a very different way, for example, than MERS, where the infection stayed on at very low level but continued. And instead, with COVID-19, we simply, because of how contagious the virus is, you know, we haven't been able. So what I'm trying to say is that having that body of knowledge of basic research in virology has been so important because you learn, but everything is different. And so this allows you to speed up the research. So the basic science that our virologists do is very important. And at ASM, we published the Journal of Virology, which is a historic, very prestigious journal in virology, and that's important. But the other aspect in all this, because it really takes a village, you need to have the drug development, as we just discussed. You need to have the vaccinology in it. You need to have the diagnostics. And I really want to give a shout out to all those professionals, clinical microbiologists who are really very often, you know, seem like the Wizard of Oz, like behind the curtain. The patient doesn't really see, doesn't interact with them, but it's the magic that happens behind the curtain where you can diagnose someone, and 70% of the infection of the doctor's decision are based on those tests, right? So there is no magic in reality. There's just like very hardworking people. So it's a whole ecosystem. And we're very proud as an association to have all these people working together at this moment. You referenced vaccines, and it's very, very interesting this, and it's true. It takes a long time, but if you think, you know, what we're very proud of is how fast this is going right now, and how the technology also has changed. Now, with vaccine platforms, essentially, you know, we can accelerate to some extent the production of those vaccines. So I'm Italian, and I see, you know, vaccine platforms essentially like pizzas, right? So you could have different bases for the pizza. You can have the gluten-free, you can have the you know, whole grain or whatever, and then you can plug on top of it, you know, different toppings. And so part of why we're able to accelerate the research is that we have now this platform, the basis, and you can use the different kinds, can be RNA, can be DNA, can be nanoparticles, but then you can customize them, for the vaccine that for the infected agent that we're dealing with, which, as Julie was saying, the fridges are empty of that because we didn't know them before. So again, it takes time, but also we need to put it in perspective, of how the technology is changing and is probably putting us in a better position than when we were only 10 or 15 years ago. But it takes time, and safety and effectiveness is really the most important thing. So until we have phase 3 randomized clinical trials that prove the safety and the efficacy of these, it will take time. But science has made terrific progress.
Yeah, and in terms of accelerating that progress as well, so I'm going to version the same question I asked to Julie. ASM had a worldwide research meeting recently. Obviously, COVID-19 was a big focus of that effort. In terms of the findings that came out of that, I understand you had some focus on some key action steps for not just vaccines, but for drugs and diagnostics as well. Any points you'd like to highlight in those findings?
Yes, that was a very exciting virtual international summit that we put together very quickly. And it goes to the point that we were discussing before, how the global community is coming together and trying to not to step on each other's toes and try not to duplicate research and efforts, but try to be synergistic rather than duplicative and learn from each other. So that was the intent. And for example, the event was organized by tackling the different components, so the drug development, the prevention, the vaccines, and the diagnostics, the three big topics. And there was a lot of conversation about the small molecules and their repurposing. For example, our remdesivir, and fascinating the mechanism of action of remdesivir, and how it is absolutely essential for the virus to have to work through the viral DNA-dependent RNA polymerase, and through this drug, incorporation of this base analog in the transcription incorporates a 3-phosphate version of this that blocks 3 nucleotides down the replication of the virus, which has been shown very effective with other coronaviruses and is showing very encouraging effects on SARS-CoV-2. However, even exciting, I learned at this summit that we had is that there is another drug in making of this, which is called EIDD, which is very similar to remdesivir, but remdesivir needs to be administered IV, while EIDD would be an oral. And so if that proves to be effective and safe for SARS-CoV-2, that of course would be a very interesting drug. Also, I could go on the science if you want to talk more about what bubbled up at our event. But one thing that became very clear is this need of curation and coordination of research, because I was just saying a minute ago how exciting it is to see all the science growing so fast in such a short time, but also it can be very confusing. And so we heard loud and clear the need for a curation in real time of the research that is produced. And so what we put up in exactly a week is a registry of a curated registry of all COVID research. And we were able to recruit 2 stellar virologists for this, Dr. Lynn Enquist from Princeton and Dr. Harold Varmus, who's now at Cornell, former NIH director and Nobel laureate, to really curate with the other set of illustrious curators the best science that is coming through right now and aggregate it in one place. So I would encourage our listeners to go on the asm.org website and click on registry and see how well curated is the key information that is needed. So this effort that we're trying to make is that of of curation of the research to make sure that we accelerate as much as possible the progress by providing the whole scientific community vetted information of what could be very relevant for drug development and vaccines and the whole field.
That sounds like a really helpful resource. Now, we've talked about therapeutics and we've talked about vaccines. Also important right now in managing the pandemic are diagnostics. And Stefano, I know that's another area where ASM has been very active. You've been involved with the White House task force. It does seem like this pandemic is one surge after another. You know, last month it was vents and hospital beds and ICUs that we had to ramp up, and looking ahead to May as people talk about steps towards reopening and a need to have a much better capacity to identify new cases, trace contacts, and contain further outbreaks. Diagnostic testing is absolutely critical. Any thoughts to add there about where we are and how we can improve our innovative diagnostic capacity?
Very big topic, Mark, and I think you hit the nail on the head at the beginning of what you were saying. Is in the absence of approved and proven therapies, in the absence of a vaccine, diagnostics become particularly important. I mean, they're always important. But if you think of the two major classes of diagnostics that you can use in this case, so the direct viral detection, so to be able to understand who has who's infected with the virus at this particular moment, and the serology, so to understand who has been exposed to the virus and may have developed immunity. These are two very, very important components that help you manage the whole situation, again, in the absence of treatments. And this is very difficult to do, as we have discussed. First of all, when the new virus hit, there are no tests, and the super fast sequencing of the virus, of the viral RNA, has been extremely helpful. And you have to think that the test for detecting the virus first can be done in different ways. And this is where, and I think we'll talk more later maybe, of public-private partnerships on this, because our system, it's typically that the private sector most likely is developing tests, diagnostic tests, which are molecular tests, typically PCR tests. And then they get deployed to the various laboratories in the hospitals that use it for diagnostic. Or there can be another kind, which are what are called the LDTs, the laboratory developed tests, which are developed by the hospital, by the high complexity CLIA labs in the hospital. And so at the beginning, we had both things happening, and at the same time, there were problems in having those deployed quickly enough. And we surveyed, ASM has a network of over 900 clinical microbiologists that are covering 300 hospital labs in the country. And by monitoring and surveying our clinical members, we were able to understand what were the roadblocks that they were experiencing in their hospital that was preventing the deployment of this testing as fast as it was needed. And it turned out that there were many issues, but one was a regulatory issue that the FDA has in place once an emergency is declared is what is called the EUA, the Emergency Use Authorization. So essentially the tests that normally a lab, a hospital high complexity CLIA lab could develop, at that point they need to get, and they wouldn't normally need authorization from FDA, at that point they did needed it because it was an emergency, and so there are commercial incentives to avoid some fly-by-night labs that could take advantage of the commercial opportunities. So, but that provided a very heavy burden, because people may not know that we have about 2,500 clinical microbiologists in the country, and we run at an about 10% vacancy rate in clinical microbiology under normal conditions. Now, superimposing this a pandemic and the needs for the lab of developing new tests and testing a lot of people, And then put on top of that, the need for dealing with the FDA authorization to get your test. It became an explosive situation for the hospital labs to develop the testing. So ASM worked very, very closely with FDA, and I have to say how wonderful it has been working with FDA, how receptive they have been of real sound advice coming from the people in the labs. And the EUA has been changed to adapt to these needs, and I think that has been a significant success. However, there's always something else, right? So now the issue became the supply chain. Essentially, the whole world does not have enough reagents that are needed and swabs for testing, for collecting samples and for testing. So as you referenced, we are now working with Ambassador Birx directly on the COVID-19 task force to try to address this issue of supply chain and how to understand, given our network of labs, what they need and where they need, and try to help coordination of these efforts to make sure that we achieve efficiencies. Because what we are hearing is that labs, for example, are using different platforms for testing, because if they run out of reagents for one platform, they can switch to another one. But that is a very inefficient way of doing testing, because you may have a high throughput machine that can do thousands of tests that at the moment you can't use, and you need to switch to something else which may not have the same capacity. So it's an incredibly complex issue. And so we're working really hard right now in see how we can use our knowledge through our members who are in the labs to help the administration as much as possible in guiding the supplies to the right labs for what they need at the right time.
Great. Well, thanks for that great summary of all of the challenges involved in innovation and diagnostics, and then a lot of logistics too. You all, I want to go to questions from. We've gotten a lot, and please keep them coming. We'll try to get to as many as we can. I want to go to questions from our participants in just a moment, but let me follow up on this issue of public-private partnership. You mentioned the White House task force. Both of you mentioned, and I completely agree, a lot of hard work by FDA staff to take on new problems as collaboratively as possible, with as much communication as possible in the development of new therapeutics and diagnostics. Julie, maybe go back to you for any further comments that you've got about the importance and the potential, and maybe some of the challenges with public-private partnerships to address the pandemic. On the one hand, it seems like they're really essential for dealing with everything from avoiding duplication and making the most of our clinical trial resources to accelerate progress on developing treatments, and to make it possible to address these complicated logistical issues at scale that Stefano was just describing for diagnostics, for example. I think that's probably ahead for some of the therapeutics and vaccine production capacity too, if we don't plan well. On the other hand, it can be challenging to get everybody to work together. So any lessons about how we can do this effectively as we move forward in trying to deal with the pandemic?
Thanks, Mark. And before I answer that question, there is one thing I just really want to say, because I might not have a chance before we close. Thanks to Research America, as well as ASM and Pharma and Bio, for all the work they're doing in the context of this COVID situation. But I also thank sort of the surround sound of people and organizations that have been advocating for research funding for a long time. IDSA, SHE, the ASTO, NACHO, the American Public Health Association, all the people who are advocating for the defense laboratories that do research that's very relevant here, the JASONs. There's so many people who have been standing strong for a long time, arguing for the investments in basic research as well as pragmatic biodefense research to really prepare us for these situations. So I would be really remiss if I didn't acknowledge that all of these partnerships are standing on the shoulders of a lot of great work going on in academia, in private NGO type laboratories, as well as within the biotech and biopharmaceutical space. And when you bring this amazing intelligence capability together to solve a relatively small set of problems, it's an awesome resource. But it isn't well organized and managed under ordinary circumstances for obvious reasons. There are a number of mechanisms that are being proposed in various domains of the world of partnerships to try to solve for this. I think one of the places where Merck is especially interested in contributing is in the NIH coalition that Dr. Collins has created, the ACTIVE coalition, to try to bring the right investigators from the private and public sectors together to say, first, what are our priorities? How do we really identify what are the best opportunities that we have to solve this problem? And how do we prioritize our research and clinical development work around those? How do we share our knowledge in creating protocols and approaches to this kind of research that can accelerate? One of the great examples of that is if you're doing a clinical trial of drug X and you've identified 1,000 people who would be willing and eligible to participate, but you only really need 300 for the endpoint that you're looking for. The other 700 people are standing by, and they should be able to immediately determine their eligibility for the next trial that's standing ready to go. By aligning the eligibility criteria and the various dimensions of how we design adaptive protocols, we can really accelerate the enrollment and the movement of people through the clinical research environment. So I think that's very critical. And then lastly, of course, these coordinating private sector public partnerships really allow us to share information in appropriate ways that don't violate any of the non-competitive requirements that we have to operate under in the private sector because of the Securities and Exchange Commission rules against anti-competitive behavior. This is the time where we need the most openness, the most transparency, and the fastest track to dissemination and publication that we can possibly sustain with good quality. So I think the private-public partnership mechanisms are maturing, but they are absolutely essential both to the integrity of the science, but also the acceleration of getting to the answers that we need and telling those of us who are responsible for the scaling of all of this when we decide the right thing to do, we've got to know where's our best bet, because these are going to be macro investments necessary to really make sure that these innovations are available to all the people who need them.
Yeah, a lot at stake. So I've gotten even more questions in. Now, some of them have been covered or at least touched on by your answers in the last few minutes, but I want to go back for a minute to diagnostics. So a lot of questions in about diagnostic tests of various types, including factors that some early science at least suggests may influence susceptibility or complications from COVID-19. But I'd like to focus on one, Stefano, maybe you could take us through the issues here, and that's serology testing for antibodies to COVID-19. I just want to emphasize that a lot of the testing we were talking about before was about testing for the presence of viral antigens, actual molecules of the virus present in nasopharyngeal swabs. It's an indicator that people are shedding the virus and contagious, that they're actively infected. Another important type of testing is for antibodies for people who've been previously exposed. There are a lot of these on the market now in the US and elsewhere based on versions of emergency use authorizations, or at least FDA allowances. But there are some real concerns on two levels. One about the accuracy of these tests. Are they really accurate enough to give a quantitative estimate that is a reliable predictor of immunity? And the other is about the underlying science. Do we understand the immunity of COVID-19 infections enough to be able to say, based on a serology test result, that someone can confidently go back out and maybe work in a job where they might otherwise be at risk of transmitting the virus or contracting it themselves? So maybe in just a couple of minutes, Stefano, big issues you see there that could be addressed through some further, hopefully rapid innovation to improve these tests.
Yep. Thank you, Mark. And serology right now is really a big issue, and we're very focused on that. Essentially, just like you said, with an antibody test, it's important to detect whether an individual has developed antibodies against the virus. However, better remind, especially for those who may not be scientists on the listening to us at the moment, IgG and IgM that are detected in these by these tests, they can be specific for COVID-19 or may not be that specific. As I said, we have several coronaviruses and some are quite similar to each other. So the specificity is very important, is the number one thing that's very important, because you don't want to have false positives, as you said. Otherwise, you think you may have developed antibodies, but that maybe not directly toward the antigen that we are dealing with right now. So that's one issue, that is specificity. The second issue that that is hard to determine at the moment is that if you have developed antibodies, that you want to know, are those antibodies actually blocking antibodies? So are really helping you to be immune from the virus? And at the moment, we just don't know. It's likely that if you have developed those antibodies, you could be protected, but we don't have enough data yet to say that by all means you are going to be protected if you have those antibodies. But having the test, which is specific and reliable, is very, very important. So we're working again with FDA. We're becoming really good friends with our colleagues at FDA. Because here we asked the FDA for some clarifications. I was just saying that in the viral test, the PCR test I was talking about before, those developed in the hospital high complexity CLIA lab. These are very sophisticated labs, and there's a general trust that what they do is actually reliable, and they do that all the time for many different tests. So in a sense, there's nothing different than usual. For the serology, though, there was a little bit of a different rules under the EUA, and there was a need for some clarification. And we're working with them in saying that if the serology test has not been developed within one of these high complexity CLIA labs, it does require EUA, and that is very important. So if you're looking to get tested or so, what I would look for, what I would tell my kids and my family and my friends, is really making sure to know that for a serology test, that either they have received the EUA from the FDA or that it's developed in a high complexity CLIA lab, or both. That, I think, is the best guarantee that we can have for having reliable testing. It's very important. And again, the goal is to try to come back to some sort of normality in the country and in the world. And as I was saying in the beginning, having reliable and accessible testing is going to be key at this moment for doing that. If we can't have reliable and omnipresent testing to be able to test people often, and very interesting conversation would also be when to test, because you don't develop antibodies immediately. So it gets very complex. But these are the kind of guidelines that are very important for people to know, to understand, and for FDA to ensure that they put that seal of approval, so to speak, that the test is reliable, because it's very important.
All right, there are a number of questions asking us to maybe go out on the limb a little bit and make some, if not predictions or guesses, but at least some informed opinions about what might be available when to help reduce the impact of the pandemic. So we're seeing cases go down now. I think there's a lot of hope that that the test and trace capacities and some continued distancing measures and the new normal that's coming could contain outbreaks in the near future. There's a lot more concern, I think, about a potential additional wave, maybe a bit later in the year, in the fall, maybe combined with the usual seasonal flu outbreak, which could be pretty challenging if we haven't made more progress on diagnostics and therapeutics. So first, a quick question, and maybe Stefano, this is for you, but either, how long till we get that reliable serology testing so that people can be confident about their immune status for COVID-19?
Yeah, good question. So I think there are many tests that are already being developed, and several have already received EUA approval. So this is good. And validation issues are very important right now, but the tests are coming. I think the big issue is going to be, again, that of scaling up right now and making sure that they become broadly available. There was just in the news today, I wasn't aware of the study of serology testing done in California by Stanford, I think, randomly testing population. Very interesting study. I don't know the details yet. I just saw it in the news today. So those type of studies done on a large scale require high capacity for testing. So I think we're getting there. I think once we have the validation and the right tests which are coming through, the big issue, as I was saying, also for the direct viral tests for the PCR is scaling, which is a big issue right now.
Scaling is the repeated one of the repeated core challenges in this pandemic. There's a new report out from the Rockefeller Foundation that proposes some important steps around trying to normalize these supply and allocation mechanisms, which just again can't be underestimated in pandemic response. Let's assume we get a good serology test soon. Julie, what's your sense about whether we're going to find enough people who have immunity that's going to be a real source of containment in the fall? My sense is that for a virus that's as contagious as this one, based on what I've seen in terms of population level immunity and in areas where there have been big outbreaks, is that we really can't count on that. Too much potential for spread of a virus that has such a multiplicative factor when people are infected under normal circumstances of interaction.
So Mark, I don't want to be an outlier here, but I'm very circumspect in thinking about testing. I think we may have created the impression across our country that somehow testing is the ticket to restarting our economy. And I think it's a little more complicated than that. I think we need to be very clear about what is the testing for. Highest priority still is testing people with symptoms who are being admitted to the hospital so that we can make sure they're getting the right care and are diagnosed properly. So that has to still remain the single highest, greatest use of the direct virus testing that we have. I think it's also important in communities where we're not seeing widespread ongoing transmission that when we see a patient who has symptoms of the disease, that we are able to test that person and diagnose them quickly and perhaps test the people they've been exposed to or around them so that we have the opportunity to locally quench the new, sort of firecrackers that could start a bonfire of spread anew in a new location. I think it's important for the CDC and other public health entities to be able to do population level testing to truly understand the full scope of kind of the iceberg that we're dealing with. We know the patients who are in the hospitals, and we really don't understand how much spread really has gone on in the community. And the danger with bad tests in that situation, they're going to misinform us or falsely reassure individual people that they either haven't been exposed or that they are falsely reassured that they're immune. I don't see that a positive antibody test right now today tells anyone that they're safe. I think we hope that that will be the case, at least for a while, that those antibody tests will be equivalent to protection from reinfection. But we don't know that, and we don't know how long that protection would last. So how often are we going to suggest that people get tested? And then finally, I hope this doesn't happen, but this virus could evolve, and the immunity to the virus circulating today might not be protective against the version of the coronavirus that's circulating in the winter or spring. So we have to have an open mind and really do the careful science before we rush out and unnecessarily waste the precious testing capacity and resources that we have using the tests for things that aren't really helpful in containing the outbreak or helping to treat people or understand the risks and dangers that healthcare workers are facing, and really get sensible and prudent guidelines around this. So I'm not one to rush out and say Merck needs to test all of its employees so we know who can come to work. We're not anywhere near population level herd immunity yet. And so that cannot be the criteria for restarting our economy. We have to find other ways to get people back working and kind of get the right balance between our social distancing measures, the protections that we'll still need to protect hospital surge capability, and above all, protect the front line of workers who are giving their hearts and souls and sometimes their lives to taking care of patients with this very difficult virus.
So it sounds like this is going to be with us for a while, that the testing strategies that you described around symptomatic people, higher risk individuals can help. Serology testing, when it gets better, can help. But with the virus is still going to be with us. There's still going to be a lot of people who are susceptible. We've talked some about the longer term benefits from vaccine availability for transforming this new normal. But in the meantime, distancing and other steps might be alleviated by progress on other therapeutics. And we've touched on some of these, antivirals that may help. And I recall the early days of HIV treatment when we didn't have great treatments, but the first ones helped some, and it provided a foundation to build on. So over time, the antivirals got a lot better. You all mentioned the immune mediation therapies for people who have very severe reactions that may be related to cytokine storm, and those kinds of drugs are in testing now and may have relatively fast results. Just to wrap up very quickly, any other technologies that you think could take the edge off the severity of a risk for a second wave and could help support this other combination of steps that we're going to need to go through until we get a vaccine? Monoclonal antibodies, others, any last words?
Well, all of the above, Mark, but I think the single most important early game changer would be an antiviral that either prevents the people who have the mild upper airway disease from progressing to the severe disease, and we know that that's a subset of people, but preventing that serious change in clinical prognosis would be huge, or something that actually could really turn around the disease in the people who have that severe lower airway disease. That's a harder thing to do, but it might be possible. And we're all looking with our breaths held about some of the data that will be emerging soon. So if we can lower the mortality rate from this disease and reduce the requirement for ICU care, that's a game changer. I mean, we could imagine a situation where we're dealing with yet the fifth cold virus. That would be a miracle. But we're not there yet, obviously. And until such time that we are lucky enough to have a therapy like that and can make enough of it to treat all the people around the world who are at risk, we have to do the things that we're doing now.
Keep up the overall strategy.
I agree completely, and actually, I want to close the loop on what you were saying before, Julie, about the testing. No, you're not an outlier. I agree with you completely. Given the testing capacity that we have now, you want to test for those individuals where the information is useful for a therapy or a cure. But the reason for ramping up is that it's going to open up so many more possibilities, in the future for what is needed. So I just want to close that. I don't disagree with what you said. And Mark, to the point that you just made, I think that I agree with Julie that the small molecules is probably one of the fastest thing that we can get to. But it's very interesting, the landscape. I don't know how you see it, Julie, but there's also several new things that are being tested out, like stem cells, for example, mesenchymal stem cells. That kind of interesting. As an approach, I don't think it's one of the fastest, but it's interesting. There's some other convalescent serum. I think it's very interesting what's happening in that space. So I think there's a very composite landscape of approaches, which gives us a lot of hope.
Yeah, so I think that's a nice note to end on in an unprecedented pandemic. So Julie, Stefano, thank you both for an excellent discussion. I want to turn this back over to Jenny to close us out, but let me end with a word of thanks on behalf of all of us to Research America. If anybody had any doubts about the value of research, basic research, translational research, clinical research to take these products through more efficient testing where they really work in people, and then applied research, including in manufacturing and even in related issues like real world evidence and ways to improve the distribution of medical products. If anybody had any doubts about the value of those investments, it's really clear here. And just again, special thanks to Research America for really carrying the torch on the needs for more support for critical efforts like these. Jenny, back over to you. And again, thanks to Julie and Stefano.
Thank you.
Well, thank you, Mark and Stefano and Julie. What a terrific session. We obviously could have gone on much longer, but I know I learned a lot, and I think those who joined us did as well. And thank you for those of you who are online. We really appreciate your joining us. We'll be sending out a recording of this, which you'll receive. So those of you who weren't able to get on exactly when we started, you'll get a recording. And then I just wanted to mention 2 upcoming webinars we have. These are sessions for our Alliance members. And if you're not sure whether your university or organization is an Alliance member, just feel free to send us an email through our website, and we will help you figure that out and get you connected. We have a webinar with Donna Cryer, who's the president and CEO of the Global Liver Institute, and she'll be talking about the impact of the pandemic on patients. And then on May 6th, we have a noted infectious disease expert, Peter Hotez, joining us. So I'm sure that's going to be a lively session as well. I am just going to end by thanking all of you from across the country who joined us. You'll see on your screen some terrific resources from us and our partners. We'll be sharing those as well. Thank you, and I hope you all have a good afternoon.
Thank you.